Good morning. I'd like to talk about pre-visual and early detection of myrtle rust on rose apple using hyperspectral measurements and thermal imagery. This is a real team effort, and you can see the people involved in it, which were a mix of pathologists and remote sensing scientists at Scion. Henning Bodenbaum from the University of Trier also contributed to this research. So just by way of overview of the detection target, the detection target itself was rose apple, and we chose this target because it's an easy to infect species with myrtle rust. And you're looking at here 2 infected plants. These were actually the worst affected plants over the period of the study, going from 2 days after inoculation through to 10 days after inoculation. The main focus of the study was on the early stages before the disease became visible, and also just immediately after it became visible. There is a paper that describes this in detail, and it is available as a first look paper on phytopathology, and the DOI for that paper is given below. So by way of introduction, myrtle rust is caused by Austropocenia sidii, and it's a serious disease that affects many Myrtaceae species, in total 480 worldwide and around 30 within New Zealand. Commercial nurseries that propagate Myrtaceae species are particularly prone to the disease, and nurseries require a reliable method for pre-visual and early disease detection to minimize potential losses. Conventional diagnosis relies on host identification, symptom observation, and microscopic examination. But this method's not always practical or cost-effective in a nursery setting. And seeing the subtle signs of the disease en masse, or over a lot of plants, is actually quite difficult, especially when it's first being expressed. DNA-based techniques have been developed but are expensive and may not always detect the disease on asymptomatic plants. And this is a bit of a problem, because you really need to know where to look to take the sample, because they're based on small sample sizes. Hyperspectral and thermal imagery provides an alternative method of pre-visual and early disease detection. And these methods work because plants subject to disease react through changes in physiology and changes in plant pigments, which may be detected through alterations in leaf temperature or reflectance from different wavelengths. As I mentioned earlier, rose apple is one of the most susceptible hosts to myrtle rust. And so the objective of this research was to investigate the utility of models that use hyperspectral data and thermal imagery for pre-visual and early detection of myrtle rust on rose apple. The experiment was undertaken in a temperature-regulated dark room with artificial lighting. We had a total of 81 plants that were allocated to a control. 30 plants were allocated to the control, and we also allocated 51 plants to the myrtle rust treatment. The reason that we have different numbers of plants allocated to the two treatments is that we wanted to maximize the number of plants that were going to get infected. As it turned out, all of them became infected. The conditions were set to 22 degrees centigrade, 70 to 80% relative humidity, and 16-8-hour day-night cycle. And you can see the plants, the control plants in the top panel, in the temperature and the environment regulated room before and after this was closed up. The plants in the myrtle rust treatment were inoculated on the 4th of November and then placed back into the enclosure under optimal conditions for infection for the next 24 hours. The optimal conditions included 90% relative humidity and a temperature of 18 degrees centigrade. Just to make sure that the inoculation process itself did not affect the affect the results, we mock inoculated the control plants with sterile solution and kept them under identical environmental conditions. In terms of the measurements themselves, we took pre-treatment measurements, and we then took them daily for 6 days from after inoculation, from the 2nd day after inoculation through to the 7th day after inoculation. We also took measurements 10 days after inoculation. The symptoms in the MR inoculated plants all occurred between 4 to 6 days after inoculation. And as I mentioned earlier, all 51 plants were infected, so it was a very high success rate. And you're looking at the 2 worst affected plants on this slide in the top 2 rows. And so, of course, because the plants became infected over a 3-day period, we had to re-express all the measurement data with respect to the day of symptoms. So each measurement was expressed as being a number of days before symptoms, either 3, 2, or 1 days before symptoms, or DBS, the day of symptoms, or the day after symptoms, either 1, 2, or 3 days after symptoms. And you can see this from the slide. If you look at the top row, this plant developed symptoms on day 4. And if you look at day 2, to the immediately left of that, 2 days after inoculation equates to 2 days before symptoms. A control plant is shown for reference in the bottom row. I hope this makes sense, and it may become clearer as we go on. We took measurements of the symptoms to detect the symptoms which only appear on the new red leaves. Visual assessments were made on both sides of the leaves, and the aim was to identify infection at the earliest possible stage. And the opposite photos show some of the symptoms coming through. No symptoms were found on the control plants over the course of the experiment or for a week afterwards. We also took physiological measurements of stomatal conductance and transpiration on all plants at the conclusion of the experiment using the Waltz GFS 3000. Thermal measurements were taken using a thermal camera, which was set up 1.2 meters from the plants, as shown in the top figure. These measurements were calibrated by targets of known temperature, and you can see these in the top photo. And the data was processed using FLIR software, and you can see this in the bottom panel. The 2 targets are also visible in this panel. Normalized canopy temperature was determined as the difference between air temperature and canopy temperature, and this was determined at the plant level. And we also determined standard deviation of normalized canopy temperature at the plant level, because this is also a useful metric. We also collected hyperspectral imagery from the plants, and I just wanted to briefly talk a bit about hyperspectral imagery because it may require a little bit of explanation. So hyperspectral imagery basically measures reflectance across a large part of the electromagnetic spectrum. And that's what you're looking at in this figure. The green line is a typical reflectance for vegetation across the electromagnetic spectrum. And if I could just focus your attention on the visible range and the bottom left corner there, you can see where the blue, green, and red bands are. So these are colors that everyone's familiar with, and you can see the reflectance for those colors. And the reason the green is higher in this case is the reason that vegetation appears green, because there is more reflectance from the green band. But there are also many other useful attributes you can get from hyperspectral imagery. If you go above the visible spectrum, it tells you information about cell structure. Even further above that, you get into the shortwave infrared part of the spectrum, which gives information about water content. And in terms of detecting disease, there are attributes from right across this range that are useful, because disease may affect water content. It can also affect pigments and structure as well. So hyperspectral imagery is very useful for telling us about infection and disease. Hyperspectral measurements were taken from both red and green leaves using a Spectral Evolution RS 5400 spectroradiometer. This is a handheld device, And we focused on both the red and the green leaves because we were wondering if they had different responses to the infection. So just to give you an example of what this data looks like, this is data collected from the red leaves, sorted according to the 8 infection stages from 3 days before symptoms through to 3 days after symptoms, and the pre-treatments also shown in the top left. The means for the Myrtle Rust and control plants are shown as thick lines, and the thin lines show the plant level variation. There's a lot of data to take in, so I just want to focus your attention on the visible part of the spectrum again and just blow this up and look at it in more detail because it is quite interesting what you can see from this part of the spectrum. If we just have a look at the top left graph, you can see that in the pre-treatment stage, the reflectance was fairly similar between the 2 treatments, but if we go right across to the right-hand side of the figure, you can see that the reflectance for the Myrtle Rust was increased around about 550 nanometers, and this is because chlorophyll was reduced at this stage, because this part of the spectrum is a very good proxy for chlorophyll content. And if we look at the BGI2, which is a ratio that's used to measure this particular phenomenon, then this is expressed as the reflectance at 450 nanometers over 550. And if we overlay the actual values for BGI onto these 2 graphs, you can see they were fairly similar in the top left panel, but on the far side, on the right-hand side, you can see that BGI was reduced in the Myrtle Rust plants compared to the control plants, which indicates that chlorophyll was being reduced. So this is just an example of how hyperspectral imagery can be used. And of course, this is just one index, but we had 101 indices like this that we derived from the data. So it's quite an information-rich data set. So I'd just like to talk a little bit about the data analysis. We developed classification models for the pre-visual stages. That's 3 days before symptoms, 2 days before symptoms, and 1 day before symptoms, and also the 4 early detection phases that include the day of symptoms and 1 day after symptoms through to 3 days after symptoms. And each classification model used 3 different groups of predictive variables that included the 101 narrowband hyperspectral indices from the green leaves, the 101 narrowband hyperspectral indices from the red leaves, and the thermal indices. So if you combine these 3 different groups of data with the 7 detection phases, that's 21 models that we developed in total. Because we had 101 narrowband hyperspectral indices, we needed to somehow reduce down the number of variables that were used for each one of these models, these 14 models that used hyperspectral indices, because you can't really use 101 variables when you only have 81 data points. So we used this recursive feature elimination, and we were able to reduce down the number of variables to between 2 to 14 for each model. Classification itself was undertaken using regularized discriminant analysis. We tried 2 other machine learning methods, but regularized discriminant analysis was slightly more accurate, so we went with that. And we used a 5-fold cross-validation with 5 repeats, and the reported model statistics were averaged across all 25 validation data sets. The calculated statistics included precision, which is the proportion of positive predictions that were correct, the recall, which is the proportion of actual positives that were correct, the F1 score, which is the harmonic mean of precision and recall, and we also used accuracy because this is a really well-known statistic, which basically gives you the proportion of correct predictions. The F1 score is most widely used metric for classification using unbalanced data sets, and our data set was unbalanced because we had 51 Myrtle Rust plants and 30 controls. And it's worth noting going forward that F1 values greater than 0.9 represent outstanding classification. So just getting to the results, this this slide shows the final classification models that have been distilled down from the recursive feature elimination. On the far left column, we have the type of data, narrowband hyperspectral indices or thermal indices. On the next column over, we have whether the narrowband hyperspectral indices were from green or red leaves. The thermal indices were actually from both because they were canopy level measurements. And then in the next column, we have the 7 infection stages for both the green and the red leaves. And for the thermal indices, we used both variables for all 7 infection stages because these were the only 2 variables available. But you can see the variables for the narrowband hyperspectral indices that were selected by the recursive feature elimination. And as I mentioned previously, these were reduced from 101 for each model down to between 2 and 14. So just focusing in on some of the variables that were included, there were a number of indices that were included in more than one model, which does tell you a lot about their importance in detecting myrtle rust. And ratio 975 and healthy index, or HI, were the most commonly used variables for the indices derived from the green leaves. DCAB CXC, or chlorophyll carotenoid index, and BGI2, which is the index I mentioned earlier, were the most commonly used variables for the red leaves. So just digging into the results a bit further, this slide shows the changes in some of the key indices with respect to infection stage for the control and myrtle rust plants. On the top level, in the top left, you can see that the normalised canopy temperature didn't really change much between the treatments until we get to 1 day before symptoms, and then there's a major drop in the canopy temperature for the myrtle rust plants, and this keeps diverging from the control right out to the very end. And this happened because the pathogen punctures the leaf surface and the plant transpires more, which cools down the leaf. If we go across to the panel opposite that one, you can see the standard deviation in temperature. And once again, this starts diverging between treatments at 1 day before symptoms, and then it continues diverging until the end. And the reason this happens is because the pathogen is just attacking discrete parts of the leaf, so you end up getting quite a high variation. Some parts of the leaf are cool, some stay at normal temperature, and so the standard deviation on the myrtle rust plants is a lot higher than that on the control plants. If we go to the next level down, we're looking at variation in healthy index, and generally healthy index declined below that of the control after the inoculation. And if we go across to ratio 975, you can see that this declined below that of the control after inoculation, but then increased again at 1 day before symptoms before leveling out with the control after that point. And finally, the bottom 2 panels, the left one shows the chlorophyll carotenoid index, and it shows that this index declined below that of the control from the very immediately after inoculation at 3 days before symptoms, and then continued declining. And we had a similar trend for BGI2 as well. Some quite marked changes occurred after inoculation. So I'd just like to show you the results from the thermal imagery. The great thing about the thermal imagery is that the 2 variables that we use can be shown on one graph because there are only 2 of them. So on the y-axis, we have the normalised canopy temperature, and on the x-axis, we have the standard deviation of the normalised canopy temperature expressed by the control and the myrtle rust plants with respect to the infection stage. So if we look at the top left panel, the 2 treatments are completely overlapping, pre-treatment and at 3 days before symptoms, and then 2 days before symptoms. But then by 1 day before symptoms, there's complete separation of these 2 groups, and this continues right through to the end. And by 3 days after symptoms, these groups are very well separated. But even at 1 day before symptoms, you can see just by eye that it's possible to completely separate out these two treatments. And this occurs because the temperature in the Myrtle Rust plants' canopy is becoming a lot cooler and more variable than the control. In terms of the overall model results, this slide shows a summary of the results, and it gives the F1 score followed by the accuracy. The models highlighted in green have outstanding accuracy. And just looking at the thermal indices, you can see that by 1 day before symptoms, there's 100% accuracy, and the F1 score is 1.0. And this continues right through to 3 days after symptoms. If we look at the green hyperspectral indices, you can see quite high accuracy at classifying the two treatments, even 3 days before symptoms. It narrowly misses out on having an F1 score of 0.9, but the accuracy of 91 is actually pretty high. And then that continues right through to 1 day before symptoms. So excellent pre-visual classification using the indices from the green leaves. In contrast, the indices from the red leaves were quite good at classifying the early detection stages, and you can see that from the 0.92 F1 score at 2 days after symptoms, which continues through to 3 days after symptoms. So it was quite interesting to see that the narrowband hyperspectral indices from the older green unaffected leaves were more accurate for classification than the indices from the red leaves. And I think this really highlights that you should study the leaves that are not affected by the disease, because there may be physiological changes that are going on within these leaves as well. And I think this was a pretty important finding from the study. The key narrowband hyperspectral indices from the green leaves were ratio 975 and healthy index. Healthy index describes changes in photosynthetic function, and ratio 975 is a water stress index, which describes variation in water content. The key narrowband hyperspectral indices from susceptible red leaves provide insight into the processes occurring in the leaves. The blue-green index has been developed as an accurate proxy for chlorophyll, and similarly the chlorophyll carotenoid index is also a NBHI, which is positively related to chlorophyll and carotenoid content. So changes in both of these indices suggest that infection induces reductions in chlorophyll within susceptible red leaves. In terms of further research, the accurate characterisation of Myrtle Rust shown here suggests a robust detection methodology could be developed in a nursery setting. One of the limitations was the sample size was quite small, so further research should be undertaken to check generality, and this is particularly the case for the hyperspectral indices. The results from the thermal imagery were a lot clearer and I think reasonably robust. This approach should be extended to other Myrtaceae species, and we will next focus on detection of Myrtle Rust on key eucalyptus species. So in conclusion, all inoculated plants developed minor symptoms that will be difficult to detect through visual observation. The validation data showed models using thermal indices could perfectly distinguish treatments during both pre-visual and early detection stages. The use of hyperspectral indices from green leaves provided excellent pre-visual classification, and these results are promising, and further research should extend and scale up this approach. I'd like to thank Scion for SSIF funding, Auckland Council for access to collect the seed. There are a number of staff at Scion who helped with the disease assessments and plant maintenance, and Scion nursery staff for propagation and care of plants. Thanks for your attention.
Thanks, Michael. That was really awesome and really interesting. I just had a quick question about, do you think that because when Myrtaceae are attacked by insects or like any sort of damage, they have that response in which they go kind of reddish, and sometimes it's even difficult to see whether it is Myrtle Rust or not. Is that something that you might be thinking about testing to see if you could decide if it's Myrtle Rust or whether it's insect damage?
Yeah, that would certainly be a challenge to try and separate out those two effects. Yes, it could be something to check. And of course, it's easy under these controlled conditions to really isolate what's going on, but certainly it's the next stage is to start to separate out multiple stresses on the plant.
Yeah, it's really, really interesting. All right, we have a question. Can we use UAV to capture multispectral and thermal images instead of handheld devices? What bands are you using?
Yeah, certainly we can use a UAV to collect thermal imagery, and that's part of our group has the capability to collect thermal imagery from a UAV, as many other groups no doubt have. So that's not a problem. Yes.
Good. So we have another question. What's the opportunity to use this technology in an automated early warning system for biosecurity surveillance, or perhaps for preemptive disease control in a nursery or field situation?
Yes, that's a really great question, and I think it's going to come down to how easy it is to collect the data and then quickly analyze the data in those sort of situations, which is what we're working towards. Possibly the standard deviation of canopy temperature may be a better metric to focus on because it doesn't require as much calibration. But certainly there is a bit of work to go in before we can automate this detection system. But certainly it's high on our priority list to get to that point.
Sounds good.
I think sorry, I'll just add to that one.
Yep.
I think nursery setting provides quite a good bridge for that because it's a more controlled setting, but it's still a realistic setting that is going to be useful to people.
There's also a second question. What causes the temperature to drop in the diseased plant?
Yeah, that's a great question, and it occurs because we think it occurs because the pathogen enters through the leaf and penetrates the cuticle, and as a result, the plant transpires more. I didn't show it in this presentation, but we have measurements showing that the Myrtle Rust plants have higher transpiration, and the higher transpiration cools the leaf, and that results in the reduced temperature.
Really interesting. Right. Sorry, lots of questions coming in now. One of the questions is that how do you think that this technology would work outside the lab?
Yeah, I think it should work reasonably well. I have read articles where thermal imagery has been used to characterize stomatal conductance in a field trial for phenotyping purposes. So I don't really see any reason why it won't work outside a lab. It's hyperspectral imagery, requires a lot of calibration, and thermal imagery requires calibration as well. So it's definitely at the cutting edge of science, but on the other hand, I have seen examples of this being applied operationally in phenotyping trials for a tree species. So I think it should work. We'll just have to work through all the potential issues and get processing pipelines in place that can efficiently process the data.
Sounds good. All right. So do you think that you could use this as a tool for seedling selection or in a tree breeding setting?
Yes, I think so, very much so. If you had a range of genotypes and you wanted to work out which are resistant to disease, it may be a way of screening en masse a lot of plants. Certainly, I think that's a really useful application of the technology, particularly if there's a high inoculum around and you know that some genotypes are not becoming infected. It provides a means of quickly assessing that. But also in field situations, we've also used hyperspectral imagery to characterize the extent of disease as well, and diseases like Dophostroma, and it's quite effective in that setting. So definitely useful for selecting clones that are less affected, for sure.
Yep. Right. Have another question. Lots of compliments on your awesome presentation. Have you tested the limits of sensitivity with this technology? I assume the concentration of the inoculum is reasonably high compared to what you might expect in nursery or native estate settings.
Yeah, that's a great question. No, we haven't yet, and you're right, it was pretty high. We did want to get the plants infected. We really undertook this as a proof of concept to see which indices are likely to be most useful, but certainly going forward, that will be an important question to ask.
That's good. And then the last question is, what is the symptom you are seeing at day 1? Looking at figure 1 online, it's not obvious, but day sim.
Sorry, do you want me to go back to that figure? I can just quickly go back if you like.
Sure, yep.
Yeah. Yes, I think they may be referring to this figure here. And as I mentioned, it was very subtle, these symptoms on many plants, and a hand lens was used to detect it. So this is really part of the reason why thermal imagery is so useful, because it can actually pick up things that are very difficult to detect. And as I mentioned, these were the 2 worst affected plants. So many of the plants, it was very difficult to see them. Luckily, we had 2 very experienced pathologists looking at the plants, so we were able to pick them up very early.
All right. Well, that's it for today. Thanks, Michael, for your presentation. So a video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days. It will also be emailed to everyone who has registered. The next webinar is on Wednesday, May 24th by Nikki Reynolds. Her talk is titled Boots on the Ground, Rahutu Preservation in Mahanihana, and it's going to be very interesting to hear about that. Thanks, everyone. Hi, Riva. See you next time.